All right, so the question is, how do we actually get all of our ATP? And if you're a human, well, the nice thing is that plants are going to actually go to the trouble of making the sugar for you. That glucose is going to be the single main compound that actually drives most of our ATP. So nutritionists will tell you you've got to eat a lot of things, but and there are reasons why you have to eat those other things. But the reason why you eat sugar, and the only one of those things that directly goes in to make a lot of ATP is going to be glucose. So yeah, there you go, nutritionists. Yeah, don't at me. All right. So let's see. We have talked about how we put together the sugar, and the plants did all that. And humans just, stick the plant in the ground, wait around for it to do its thing, and then hack it down and roast up the delicious corn that was inside, or wheat, or rice, or whatever. Yeah, that's kind of who we are, filthy, scavenging parasites of plants. And there are several processes here that we could learn all the parts of. So glycolysis is going to be the one that we are going to cover, and that's going to show us how we actually break down the sugar molecule and start to make the ATP. Most of the ATP, though, is going to be made actually inside the mitochondrion, and we're going to have things like the acetyl-CoA is going to be made. We're going to have the citric acid cycle. This is going to indirectly drive the production of fatty acids. We're going to have electron carriers involved, and eventually we're going to make a whole bunch of ATP over here. But for the purpose of this class, that's all good stuff to learn. I just figure we have to focus on some part of this. We probably can't get it all in there, although it is all good and useful and real legitimate biology. So we're going to focus on the glycolysis side, and I think we can show you some fairly simple ways that'll help you remember what these molecules look like. But we're going to go into a little bit of extra detail. This is going to be the one where you actually draw out the molecules and show how they change. Again, it's just going to be the carbon backbone of the molecules and the phosphates, rather than all the other little side groups like oxygens and hydrogens. And that's going to be enough for you to get the idea of how the process works. So that's what we're trying to build here, some little bit of biochemistry, which is, yeah, frankly, the stuff that keeps us all alive. So pretty important. Okay, here is the test answer in a nutshell, but I'll make a second video where I break this down bit by bit and show you how we can sketch this out and how we can remember it. It's going to involve a little thing here. So we got our glucose, which is a little molecule, and it's going to have this carbon up here. And so we can draw this as like a little hexagon with a little O in there and a little carbon hanging up there. And so I think of this glucose as being like a little dog. So that would be its little eyeball right there. And this is its little tail sticking up in the air, like dogs do, yeah, when they're happy. And so the first step is going to be, we're going to take this 6 carbon sugar. So 1, 2, 3, 4, 5, 6. Because remember, at any place where there's an angle, if they don't show you what atom it is, it means it's secretly a carbon in there. This is a trick that organic chemists use so they don't have to keep writing C all day. Because, yeah, eventually, most everything that they've got, it's got carbons as the backbone. So we're only worried about the side chains. And so in your case, we are not going to memorize which has a hydroxyl, OH, or which has a hydrogen. So imagine these things just missing. So you've just got the little hexagon with the oxygen in there and the carbon sticking up at the end. First step is going to be to break an ATP over it. So again, disaster. We are losing energy by the process of breaking down sugar. And thankfully, we're going to get all that energy back and then a whole lot more. But yeah, that comes in the payoff phase. Right now, it's the investment phase. So yeah, just like they tell you in business class, you got to invest first before you see a return. And so our investment is we're going to take an ATP, we're going to pull off the third phosphate and attach it to the tail of our dog. And so imagine our dog with an extra little thing on its tail. I think that's kind of like a poodle with one of those little fluffy balls at the end of it. Adorable, right? We are not going to have to memorize this enzyme, the hexokinase, although it's a 6-carbon sugar, so that's where the hexo comes in. And a kinase is our generic name for an enzyme that adds a phosphate onto something else. And so 6-carbon sugar, add a phosphate on, you can't call that enzyme anything but hexokinase. And now that we have our little poodle here with a phosphate on the end, we are going to use another enzyme that we don't have to know. It's phosphoglucoisomerase. And what that's going to do is, and this isomerase just rearranges the parts that you've already got there. And so we are going to rearrange the parts, and we're going to have a 6-carbon sugar still. We're going to have 1 carbon here, 2, 3, 4, 5, 6. So we've rearranged it so that our oxygen is now going to be in the middle. And this one to me looks like maybe a little house. And so you think you got a dog, you take it to the groomer, you get it turned into a poodle. That's nice. I guess that's how dogs work. And then what's next for the dog? Why you gotta give it a nice dog house. So yeah, sure. Dog, poodle, little dog house. And we're going to have 1 phosphate on the side of that. I don't know if that's like a little flag on the side of the doghouse or something. Yeah, you got to make up something there. Then the next thing we're going to do is we're going to be so happy that we've got our dog and we put it in our house that it's going to be the cheerleader pose is what I call this. And so we've got 1, 2, 3, 4 carbons in the bottom. We still got our oxygen in the middle, so it's a 5-carbon sugar that looks like a house. And we've got a phosphate on this end that was already there. We've just rotated this around like that. So it's pointed out. And we're going to put a phosphate onto the other end. And to do that, we're going to break another ATP. So we're going to lose another ATP. The enzyme that does that is going to be phosphofructokinase. And I should have said, we are going to need to know the names of all the molecules involved here. So this is glucose. Yeah, you know glucose. When we add a phosphate to carbon number 6 of glucose, it becomes glucose with a phosphate on carbon 6, or glucose 6-phosphate. That's actually not that bad. And once you know the name, you can draw the molecule. And once you can draw the molecule, you can kind of figure out what the name is. Now, fructose has the same number of carbons, 6 carbons, but it is arranged with a little 5-member ring. So we have turned a glucose 6-phosphate into a fructose 6-phosphate by just rearranging where we're actually joining up. Rearranging where that oxygen that connects the two sides is going to be located. And so, yeah, it's still got a phosphate on carbon number 1, 2, 3, 4, 5, 6. So it is fructose 6-phosphate. Why isn't it fructose 1-phosphate? Because you could call that the first carbon. Well, chemists have a particular system. And in the particular system as applied to this molecule, that is the 6th carbon, and this is the first carbon over here. So, yeah, you know, go blame the whatever International Chemistry Society for that, I guess. But it's fine, because the next thing we're going to do is we're going to clear up the ambiguity by adding that second ATP on. And so now we have a phosphate on carbon number 1 over here and carbon number 6 over here. And so that's still fructose because it's arranged like a little house. But it's now got 2 phosphates, bisphosphate, on carbons 1 and 6. So it is fructose 1,6-bisphosphate. And there's not really anything else that it could be. That's the only name you can give a molecule like this. All right. So the next step is, so we got our dog. We took it to the groomer. We put it in a little dog house. And then we cheer. I imagine it's like a cheerleader holding up 2 pom-poms. That's what the 2 phosphates are on the side. And so, yay, we did it. We did it. Next thing we're going to do is, unfortunately, we're going to rip the fructose 1,6-bisphosphate in half. Divide it right down the middle as if there was a little zipper right here. So we'll just unzip it into, I don't know, 2 half cheerleaders with a pom-pom attached. Yeah, neat. The enzyme that does this is the one we're going to memorize for the top part. It is called aldolase. And aldolases are going to split a big molecule into 2 smaller molecules in this particular case. Yeah. So aldolase. Yeah, remember it. In my mind, yeah, this is like Uncle Aldo, you know, on a car trip. And just imagine that's your uncle's name, Uncle Aldo. And the kids are being too rambunctious in the back. They're cheering. They're talking about dogs and stuff. And, you know what to do. When 2 kids are fighting in the back of the car, you say, split it up. Opposite sides of the car. That's what Uncle Aldo does anyway. Yeah. So, yeah, if that works for you, great. If not, you just come up with your own way to remember this. You don't have to remember it my way. My ways are stupid ways. You can do much better. But yeah, if you don't feel like doing much better, this will work. This will be okay. So glyceraldehyde 3-phosphate is there. This is ready to go into the next side right over here. And do you remember glyceraldehyde 3-phosphate? That's the same molecule that we were generating in photosynthesis. So if we were halfway through photosynthesis and we had made one of these molecules, we could feed it directly into step 6 and cash out a little bit of ATP. See, we're going to get some ATP back in this step. So we'll get there, though. The other molecule that you get is called dihydroxyacetone phosphate, or DHAP. And I'm okay if you abbreviate that one, because we'd have to know where all the oxygens are and all the hydrogens are for that one to make sense. So the glucose and fructose, we know it's a 6-member ring and a 5-member ring with 2 little things on the side. So those are pretty easy, but DHAP, you can just remember here. And the neat thing is that there is an isomerase enzyme that you don't have to remember, but it will turn DHAP into glyceraldehyde 3-phosphate. And isomerase basically rearranges what you've already got, rearranges those deck chairs just like this isomerase did up here. And so the isomerase will shift all of these over to this camp eventually. Okay, second half of what you would be diagramming. So in your test question, you would either be told, give me the first half of the process from glucose all the way down to glyceraldehyde 3-phosphate. And so you'd have to tell me about DHAP turning into that. But yeah, basically it's just what we covered. The other question would be, okay, tell me the second half of glycolysis, which is going to start with glyceraldehyde 3-phosphate coming in, and it's going to end up with pyruvate. And so glyceraldehyde 3-phosphate has 3 carbons on it. Pyruvate also has 3 carbons on it. That's neat. So we didn't lose or gain any carbons, and we just split our 6-carbon sugar into 2 3-carbon sugars. That's the big thing that we've accomplished with the top half. But let's see the bottom half of this. And then we'll go downstairs and I'll diagram this out on the board and show you. Yeah, this is also something that you can remember. So you're starting with glyceraldehyde 3-phosphate. It's got 3 carbons, it's got a phosphorus on the end. That's all we need to know for the purpose of this class. Next, we have a wonderful enzyme. And this wonderful enzyme is going to do a marvelous thing for us. You know how every time we've wanted to attach a phosphate to a molecule, we've had to burn an ATP in the past? It was this way in photosynthesis. It's been this way in glycolysis so far. Well, this molecule called triose phosphate dehydrogenase, and triose means 3-carbon sugar. The tree is tri for 3. Ose means it's an ending we give to sugar. And phosphate, so a 3-carbon sugar with a phosphate on the end, why, that's glyceraldehyde 3-phosphate. So yeah, it just happens to work on other 3-carbon phosphates as well. And it's a dehydrogenase, and so great. It's going to pop off a hydrogen, and in place of that, it's going to stick on a phosphate molecule. So we're going to do this twice because we made 2 glyceraldehyde 3-phosphates. One of them started out as a DHAP and turned into a glyceraldehyde 3-phosphate. The other one starts as glyceraldehyde 3-phosphate. They will both enter down here, and so that's why you got 2 of everything happening. So we're going to take 2 of those molecules, glyceraldehyde 3-phosphate. We're going to add 1 phosphorus to each of those. We'll add 2 phosphate molecules, not phosphorus, but phosphate, which is PO4. And in the process of doing this, not only do we get phosphate attached to both ends here, but we're also going to store some electrons. So we're going to take these NAD plus molecules, and we are going to add electrons to those so that we can actually turn them into NADH. And that's great. That is fantastic. We'll do that with some of the H plus that's lying around. NADH is stored energy, and we will use this later on in the cell. This drives other processes that we're not going to cover in detail in Bio 1, but it is an important electron carrier and a form of stored energy, carrying one of those higher energy electrons, rather. Okay, so now we've got our glyceraldehyde 3-phosphate. We added a second phosphate to the other end of it, And so what else can you call this? So it's glycer, because it's 3 carbons. It has 2 phosphates on it now, so it's got to be a bisphosphate. And the carbons are going to be at position number 1 and 3. So this was position 3, and we added 1 at position 1. So it's 1,3-bisphosphoglycerate. That's the name of the molecule. And again, in chemical terms, that describes exactly what it is. And if you know the name, you can make the molecule. And if you know the molecule, you can make the name. So when we're drawing this, you don't have to draw the double bonded oxygens over here, or this hydrogen and hydroxyl group on the middle carbon, or the hydrogens on the bottom carbon. We just need the carbon skeleton, and we need to know where the phosphates are. So the next step here is we are going to actually regenerate some ATP. And since we have 2 of these molecules, we can regenerate 2 of them. We're going to do this by taking one of the phosphates off. And so we're going to take off that phosphate that we just put on up here. And we'll do it in such a way that we generate ATP, and we're going to leave an oxyanion up here that's just going to hang out. That's going to stay there all the way through to pyruvate. And so don't worry about the oxyanion. We just need to draw the carbon chain. So 1, 2, 3 carbons with a phosphate on number 3. We took the number 1 phosphate off. And so this is now 3-phosphoglycerate. The difference is that instead of having C double bonded O attached to an H, we're having C double bonded O attached to an O minus. Yeah, that's the giant difference. But in terms of the carbon structure, the way you would draw it would look exactly the same as the way you would draw this if you're just drawing 1, 2, 3 carbons and 1 phosphorus. 1, 2, 3, and 1. Okay, fine. Then we're going to go through a step, and this is a mutase, and a mutase is going to be kind of like an isomerase. It's just going to change the position of something, but it's not really going to add or take anything away from a molecule. And so we're going to move that phosphate toward the center. So think of this as making and breaking symmetry over and over again. 1, we bring in a molecule that is asymmetric. Oh, terrible. It's got a phosphate on one end. First step, we're going to add a phosphate to the other end, and bam, we've got symmetry. And we can relax. But then, of course, we cash out one of those phosphates to make an ATP, and so now we're asymmetrical again. And so let's go back to symmetry. Now, it's not perfect symmetry. What it is a 3-carbon chain with the phosphate attached in the middle. And so if you don't draw the oxygens or the hydrogen or this H2OH down here, it'll look symmetrical visually. And I think that's a nice little visual clue for how to remember this, at least at the level we have to for this class. If you're trying to do this in all of its detail for a biochemistry class later on, you'll have to modify the way that you remember this. Anyway, next step is we've got an enolase, which is going to come through, and it's going to actually pop out a water molecule from each one of these things. So we were a 3-phosphoglycerate, because we were just on the 3rd carbon, and we only had 1 phosphate. We moved it up 1 carbon, so now what else can you call it but 2-phosphoglycerate? And so it makes sense, and it helps you tell the molecule from the name or the name from the molecule. Enolase is going to do its bit, and it's going to take out a water molecule, and the actual carbon structure is not going to change. So we'll still have 3 carbons with a phosphorus on the middle carbon. We just lost an H2O molecule from the bottom carbon and made a double bond in here. All right, and so the last step in this process is going to be done by pyruvate kinase. It's another enzyme that we don't need to remember. But it is going to take off this last phosphate, and it can only do that if it's dealing with PEP. Now, phosphoenol pyruvate, where have you heard that name before? Do you remember those? Do you remember PEP carboxylase? Yeah, that's the thing that makes C4 plants C4 plants. So C4 plants can take this molecule, phosphoenol pyruvate, PEP, and they can add 1 extra CO2 molecule to the end of it. That's what they do. That is a C4 plant, and that's what PEP carboxylase does. And so that's why PEP carboxylase was really easy to move from one plant to another. You just have to add the enzyme because every plant already has phosphoenol pyruvate floating around, because every plant, in addition to making sugar, has to break down sugar in order to get ATP. All right, so there you go. That solves that little mystery. And then pyruvate kinase over here is going to take off that last phosphate, and we're going to be left with a plain old 3-carbon chain whose name is pyruvate. And yeah, pyruvate is going to go on and do wonderful things inside the mitochondria. But for now, we have reached the end of glycolysis proper. So for the second half of glycolysis, I'll say take me from 3-carbon molecules until we get to pyruvate. And you'll have to know that the 3-carbon molecule means glyceraldehyde 3-phosphate. Then you'll take me through the steps and show me where we're going to get our ATP back and where we make the water in there. And you're going to tell me the name of this one enzyme, triose-phosphate dehydrogenase, and that's going to be a perfect answer for the second half of the glycolysis question. And you'll have one or the other on your version of the test. Both questions will be out there for somebody. Okay, so just a little bit of extra background to kind of cement this in and show you what's really going on there. So this is a molecule called NAD. And over in the form, you can see on this side, it is NAD plus because it's carrying a positive charge. And you see it's got a double bond here. That's how it's doing this. Over here, you can see it is just NADH, and it's picked up an extra hydrogen down here. And we've lost that double bond, and we've rearranged these other 2 double bonds so that they're over here. And so we have gotten rid of our positive charge. And so in the net, we've added 1 H+, and we've added 2 electrons, one to join this H+ onto the carbon down here and get rid of a double bond, and the other one is going up here and actually turning that nitrogen from nitrogen with a positive charge to just plain old nitrogen, neutral nitrogen. So NADH is the form that is storing helpful energy, and the way I remember it is that if it's got an H on the ends, like NADH or NADPH, that's the helpful version. That is storing energy for us. And NAD plus is going to be something that needs work. Just like P680 plus or P700 plus is a thing that's basically a problem that we had to add electrons to. Yeah, if you see it there, that's what's going on when we're talking about electron carriers like this. Okay, overall overview of what's going on here. So in our energy investment phase, we are actually going to use up 2 ATP molecules. And yeah, we're gonna just burn energy to start to break down glucose. But in the payoff phase, the second little phase here, we are going to get all that energy back and then some. So we lost 2 ATPs, but since we can do this one twice, because we've got 2 3-carbon sugars here, we actually get 4 ATPs back, so it covers our losses and adds 2 more. And we're going to get 2 NADHs out at the end. And we're also going to get 2 pyruvate molecules that aren't particularly high energy, but we can build things out of these. They have further uses inside the cell. And so the net is going to be that we're going to get 2 pyruvates, 2 water molecules, 2 ATPs, 2 NADHs, and 2 H pluses out of this entire process. And all of those things are going to be useful down the road for some purpose. So we'll have stored energy, and we'll have some 3-carbon compounds that we can do things with. That's the idea. All right, and so that's just this little green box of the process. And then there are so many other steps in the process. And I'll just touch on one of the other steps because we haven't got to the reason why you actually breathe oxygen yet. So plants need to get rid of oxygen. That much is clear, or else it's a byproduct of breaking down water, and it's also something that can literally poison them by doing photorespiration. There we go. Boy, that word always sticks in my brain. Photorespiration. Yeah. So anyway, just in case you're curious, this is the other thing that's going to happen. So the glucose is going to come in. We're going to break it down into pyruvate. The pyruvate is going to be converted into acetyl coenzyme A, and that's fine. And then that will go around the thing called the citric acid cycle. That's another cycle we could totally learn. We're not going to learn it, but a lot of biology books would have you learn it, and whatever. I don't know any point in learning all of these things. As an example, it's worth learning, I think, 1 or 2, but there's no real way to learn them all. Biology is essentially a huge place, and it's divided up into a bunch of little areas. And in my life, I've never had to actually memorize the citric acid cycle, for any of the things that I've done. And so, yeah, we're not going to do that. We just picked glycolysis as a good example. And that is something I've had to use quite a lot because the enzymes that do glycolysis are some of the enzymes that are common to every living thing, every plant, every animal, every bacteria, and every member of the archaea. So yeah, that's where they come in. Okay, if you're curious about some of these other molecules, this is a thing called coenzyme A. This is going to be the next step and the eventual fate for those 3 carbon sugars. We're going to attach them onto coenzyme A. And look at this big, beautiful thing. It's really just the amino acid cysteine shown over here. It's got a little sulfur on the end of it. And so that's one of the regular amino acids. And it's got some phosphate in the middle. Great. 2 phosphates here, 1 phosphate on the bottom. And then over here, you've got essentially an ATP that's rearranged. So instead of having all 3 phosphates in a row, you got 1 attached to the bottom and 2 attached up here. And that's the regular adenosine base. So yeah, it's a cysteine amino acid attached to an adenosine base. Neat. And rearranged somewhat. That is coenzyme A. So there you go. That's a neat thing. Here's the citric acid cycle. If you wanted to see what's going on in here, we're going to bring in a variety of 2 carbon sugars. We're going to actually lose one of those carbons to another reaction outside of the mitochondrion, but inside we're going to do this, and we're going to spin this process around, and it's going to generate, look at that. It's going to net us 1, 2, 3 NADH molecules, very helpful. It's going to net us an FADH2 molecule, extremely helpful, and we are going to get an ATP molecule out of this, but for the cost of a GDP turning into a GTP. So we basically net 1 ATP out of this process every time we spin the cycle. And every time we put in another little leftover piece of pyruvate, we can drive the cycle around one more time and get a little bit more energy out. So that's part of the reason why mitochondria are so useful. So remember we said FADH2 is one of these molecules? Let's see what it looks like. So yeah, this is FAD plus over here. And we're turning it into FADH2 by adding extra hydrogen. So here's our little hydrogens, boop and boop. And we're going to actually add 2 electrons in that process to join those hydrogens up to the nitrogen so that they can still be bonded over here. They don't have to share that extra electron. And they can bond up here. And so, yeah, FADH2 is, again, a form with stored electrons. And you can tell the H at the end means it's helpful. There we go. Last bit, why do we breathe? And again, this is just so that you know why we breathe. There is one particular protein complex called complex 4 involved in your mitochondrial membrane. The purpose of these complexes is going to be to pump a lot of H plus from one side of the membrane to the other. And we're going to build up all that H plus on this one side through an electron transport chain, so kind of like we did in photosynthesis, only it's going to involve a lot of proteins that are called cytochromes here in the mitochondrion. And then the net result is going to be we're going to have a lot of hydrogen on this side, not much on this side, and we're going to have that ATP, which is going to turn around like a little turnstile. And as it does, it is going to ATP synthase, rather, enzyme. It's going to take ADP and phosphate and smash them together and make an ATP molecule. And so we're going to net. This is where we get most of our ATP from in the cell. The oxygen is just used over here, and it's used as a place where we can dump our leftover electron. Because you remember oxygen. It's always after electrons, always wants another electron. And so this is a good place for us to dump a fairly low energy electron because oxygen will totally take it. And so we're going to take H plus and we're going to take oxygen. We'll take 2 H pluses and half an O2, and we're going to make an H2O molecule. And so we're going to use up all that H plus that's on the outside, pump everything that's left onto the inside. And yeah, this whole process is going to basically end up doing the opposite of what we did during photosynthesis. So during photosynthesis, you remember we broke down a water molecule to make an oxygen molecule. And in human mitochondria, in all animal mitochondria, or even all plant mitochondria, we're going to take some of that oxygen and turn it into water again. And so the net result is going to be that we are actually going to produce water once again, and so water will remain roughly constant. I guess if there's more photosynthesis happening, we'll have a balance. We'll make more O2 and have less water around. If there's less photosynthesis, we'll have less O2 and we'll have a little more water around. So there you go. If you wanted to see the enzymes that do some of this process, this is the one enzyme that actually is the one that uses up oxygen. It's going to break down O2. This is the only enzyme that's going to need oxygen in order to do what it does. So all that breathing that you do every single day, it's just for the benefit of this one enzyme. And this one enzyme is just involved in this one electron transport chain in the mitochondria that drives ATP production kind of indirectly by pumping hydrogen ions across the membrane. Here's a little graphical interpretation of what this is, and we actually have the video showing this molecule in action. It's a molecular animation, but it's quite accurate based on the protein structures. And so, yeah, you can see that, and it's just like a little motor. It's got a part that stays still, called the stator, and it's got a part that goes around, called the rotor, just like you would find in a mechanical motor. And we're going to load hydrogen on here. That's going to be the force that actually drives this around. When a hydrogen makes a full loop, it's going to come out this little pipe in the protein. And as every time we've spun around once, we're going to smash an ADP and a phosphate together and make an ATP molecule. So that's the idea. Here's a little picture of that electron transport chain that's going to take place inside the mitochondria. And we're going to use the oxygen as part of this one. Every time you see a site, that's going to be one of these cytochrome proteins. These FES proteins are going to be iron-sulfur cluster proteins. And there were also some of those involved in photosynthesis. So respiration, the use of oxygen and electron transport chains to drive ATP synthesis is actually kind of similar to the process of photosynthesis. They are analogous in some ways. Even the group at the center of a cytochrome is a heme group, and a heme group looks very much like the business end of a photosynthetic molecule, a chlorophyll molecule. So the net result of this process is that we're going to get about 2 ATPs out for running glycolysis, plus those other products, remember the NADH. We're going to get another 2 ATPs and a whole boatload of NADH and FADH2 out of here from the citric acid cycle. But by driving both of these processes and using oxygen, we can get another 26 to 28 ATPs out of the mitochondrion just by running those hydrogen ions through the ATP synthase and having it spin around. And so net result is going to be somewhere between 30 and 32 ATPs for every glucose that we break down. And that's a lot of energy. That's why we eat glucose. That's why we farm plants and eat them, so that we can have nice glucose to break down to make ATP. All right, and that's the end of this little production. We talked about respiration, the reason why we breathe, and the actual test question that's built into here is going to be that glycolysis, either the first half of glycolysis up to the point where we have glyceraldehyde 3-phosphate, or the second half of glycolysis where we go from glyceraldehyde 3-phosphate down to the 3-carbon sugar pyruvate. Thank you very much. That's the end of this lecture.